Hi everyone, today's our topics of discussion is MHC class 1 structure. The picture I have already drawn in my whiteboard is all about MHC class 1 molecules. So here is a cell and outside of this cell there is interstitial space and our desired MHC class 1 molecules. So first of all you have to know where these molecules come from. There is mainly heterodimer of MHC class 1 molecules, meaning one part came from one gene and another part came from another gene. So this part is mainly contained of alpha 1, alpha 2 and alpha 3 domain of alpha chain, and there is associated molecules which is known as beta 2 microglobulin. This alpha chain is mainly come from MHC class gene chromosome number 6 and it is from short arm known as P, means petite. French language petite means short, and the structure another is beta 2 microglobulin, which is not the MHC class gene, which is mainly a single gene come from chromosome number 15, but it's not from short arm. Now this time it came from the long arm of chromosome number 15. So as you seen here, between alpha 1 and alpha 2 domain there is a space known as antigen binding groove or antigen binding cleft. This part of this molecule is varying from molecules to molecules and person to person, but you have to know that there is another part known as alpha 3. This alpha 3 domain is unique and identical for all cell. So this part is mainly responsible for the interaction between effector T cytotoxic cell, which is mainly CD8 positive T cell. That's why you have to know that MHC class 1 molecule is restricted for cytotoxic T cell, that is CD8 positive. This part is known as membrane distal extracellular domain and this part is known as membrane proximal domain because it's near about the cytoplasmic membrane of our cell, and this part is transmembrane domain. That means it's a membrane spanning molecules, and here is the short cytoplasmic stress of tail, which is mainly convey the message that is outside from the cell to inside from the cell. So where the beta 2 microglobulin come from, you already knows that, but one of the main function of beta 2 microglobulin is if there is no beta 2 microglobulin with the association of MHC class 1 molecules, there may be no MHC class 1 because of beta 2 microglobulin helps the proper folding and processing of MHC class 1 molecules, and it is non-covalently bind with alpha subunit, and it is similar with structure and the organization like alpha 3 molecules, and it is not membrane binding molecules, it's just a secretory molecules from another gene. As you known before, it's come from chromosome number 15. So the whole structure is mainly responsible for the identification and distinguish between self and non-self antigens, which is mainly synthesized by in our cell known as endogenous molecules. So if anything happened wrong, our immune system like NK cell, if they don't find MHC class 1 molecule outside of our cell, they maybe think this cell is infected or something happened wrong inside the cell. That's time those NK cells kill our own cells. Sometimes some virus try to tick our own cell, so they downregulate our MHC class 1 gene, but our immune system is more clever than the virus, so NK cell don't find the MHC class 1 molecules on the surface of the cell and it thinks it is a foreign cell and I should kill it. So on that time the NK cell make the upper hand and kill the cell. One more thing, that when a tumor cell grown out of control, on that time lots more of mutation or mutated protein found in the cell, and our MHC class 1 molecules just shows the mutated protein, and our cytotoxic T cell or CD8 positive T cell find this is not the proper antigen which I want, so it's not myself, it from outside or mutated protein. On that time TCR bind with this antigen binding groove and CD8 co-receptor bind with this alpha 3 domain, and it's just release its granzyme and perforin and kill the cell. So that's the necessity and that's the structure for MHC class 1. Our next class we will be discuss about MHC class 1 processing and presentation of antigen. Thanks for